Hello everyone. So today we are going to discuss a topic called acquired immunodeficiency syndrome. It is an immunological disease. So as we are studying this unit that is immunity, so here we are studying acquired immunodeficiency syndrome. So before going to that, as you all know that acquired AIDS is caused by a virus called human immunodeficiency virus. And coming to its epidemiology, so like about 2.5 million new cases were going to be added every year, and especially in India. In India, most of the cases are seen in Maharashtra and Tamil Nadu. Like near about 50% of all HIV positive cases were from this region, and also from northeast state of Manipur, it has 8% of all the cases were seen. So this is all about epidemiology of AIDS. And then coming to etiological agent is nothing but causative agent. So what is the causative agent for AIDS? AIDS is caused by HIV virus, that is human immunodeficiency virus. So human immunodeficiency virus, it is a type of HTLV, that is human T cell leukemia lymphoma virus. So it is a type of HTLV family. Okay. Again, this HIV virus is of two different species, that is HIV-1 and HIV-2. And mostly in Asian, like US and Central Africa, it is caused by HIV-1, and in West Africa and in parts of India, it is caused by HIV-2. So both these HIV viruses 1 and 2, these were zoonotic. Zoonotic is nothing but zoo means animals. So this virus is originated from animals, especially chimpanzees. So it was found that this virus was seen as a natural reservoir of chimpanzees. Why? Because it acts as a, so this virus, whatever it is there, it will be like it does not cause any infection to the chimpanzees until and unless when that gets transmitted from these chimpanzees to human beings. Once it enters into the human beings, it causes disease. So these chimpanzees act as natural reservoir of HIV infection. So coming to the morphological appearance of this HIV virus, how does it appear? So this HIV-1 virus is a spherical in shape, that is round in shape, and its size is 100. It is spherical in shape, and its size is 104 to 100 to 140 nanometers in size, and it has a core proteins called P24 and P18. So these two were the core proteins, and also a single stranded RNA and RNA transcriptase in the core. And surrounding this core is a bilipid membrane, double layered lipid membrane is seen. And on the surface of this double layered lipid membrane, there are surface proteins called GP120 and GP41. Okay, so this is the structure. It has P24 and P18 as a core proteins, and reverse transcriptase enzyme and two strands of single stranded RNA were there. And it is surrounded by double layer of lipid membrane. So, and then this membrane is having two surface glycoproteins, that is GP120 and GP41. So these the important genes that code this virus include three types of genes, that is GAG, that is group antigen for core proteins, and POL, that is polymerase, is a gene that code for reverse transcriptase. ENV is the envelope for the envelope proteins. So these are the genes, important genes which code for these components of this virus. So these were the components, these were the genes. So these genes will code for these proteins. So these genes and viral components act as markers for the diagnosis of HIV infection. That means if we go and if we are conducting a test, and if you find the presence of these genes or presence of these viral components, it indicates this presence of HIV infection. So that is the reason these were called as markers for the diagnosis of HIV infection. So this is the structure. You can see these surface proteins, core proteins, two strands of single stranded RNA, reverse transcriptase enzymes, and then double layered lipid membrane and surface proteins GP120 and GP41. And now coming to routes of transmission. So generally the transmission of HIV takes place by 5 different routes. So it includes first and foremost the sexual transmission. So near about 70% of the populations in of the affected HIV people, they are like there the transmission mode of transmission is 75%, and it's seen mostly in like US and also in Africa and Asian countries, and seen both in bisexual and monosexual females with heterosexual propensity. And the tendency to spread this HIV infection is more from male to male and male to female compared to the spread from female to male. So there is more tendency of spread from male to male and male to female. So this is what about sexual transmission. And the second is transmission via blood and blood products. That means if you are having any injury, so you have to be transfused with some other blood components. And if at all, if whatever the donor blood, if it is having HIV virus, then it causes the HIV to the recipient. And not only that, and it also spreads by in hospitals. So it also spreads by like sharing of needles, syringes, and also etc. And it is seen in IV drug abusers. So some people will be like drug addicts, they'll be taking continuous daily, they will be taking IV drugs. So in that kind of people, if they are sharing their needles and syringes, at that time they are also at risk of getting this HIV infection. And hemophiliacs means those who require continuous transfusions, regular transfusions, blood transfusions. That includes thalassemia patients and who are like chronic anemic patients who are and also dialysis patients. So who are like these are the people who require continuous or regular blood transfusions. They are also at risk of getting this HIV virus. And then third is perinatal transmission. Perinatal transmission, that means spread of any infection from mother womb to the fetus, that we call it as perinatal transmission. And this transmission can be done through transplacentally from placenta from mother's wombs to the fetus, and also immediate postpartum period, immediately after delivery, through contamination with maternal blood. So even if the fetus is not getting this through transplacental transmission, but immediately after delivery, due to because when it comes in touch with this maternal blood, that is blood of mother or amniotic fluid or breast milk, so in that condition also there is a chance of getting transmission. So that is what perinatal transmission. And then occupational transmission. See, so many healthcare workers who are working in laboratories and blood banks, they are at risk of getting this. Why? Because they will be continuously exposed to these kind of blood products. So, if at all, I think if a person came for testing of serum electrolyte (CBP), we don't know whether he is having HIV or not. So, will be when that sample is being tested, accidentally, if a part of that sample gets spilled on his clothes or any table, there is a chance of getting a transmission of this HIV virus. And also those are the staff who are working for cleaning and all, removing the garbage and also already used needles, whatever it is that, if the waste of shops or whatever it may be, when they are cleaning that disposal of waste, at that time they are also at risk of getting this transmitted. And then transmission by other body fluids. And this HIV virus is not only found in blood and blood, it is also found in other body fluids like semen, cervical secretions, vaginal secretions, tears, sweat, saliva, CSF, synovial, pleural, peritoneal, pericardial fluid. So in all the body fluids, the presence of this virus was found, and it was found that the transmission may takes place through any one of these body fluids. Okay, so is there any chance to prevent this HIV virus? Yes, of course. So this contamination with whatever, like the healthcare workers who are working in laboratories, they are at risk of getting this. So in order to prevent the transmission, in order to keep the area sterilized, there are some chemicals which acts as germicides and will kill the virus, and it also prevents the transmission. These include sodium hypochlorite, it's also called as chlorine bleach, and 5% formaldehyde, ethanol 70%, glutaraldehyde 2%, beta propanol lactone. See, these are the chemicals which helps in killing the virus. And also it was found to be that HIV virus is very sensitive to heat. That means when it is exposed to the temperatures near about 56 degrees centigrade for 30 minutes, then the virus will get destroyed. So this is the so HIV virus can be destructed either by using these chemicals or by keeping it at a temperature of 56 degrees centigrade for 30 minutes. Okay. So this is all about the transmission. And now coming to pathogenesis. So here in this picture you can see this. So this is the virus. This is the host cells CD4 T cells. So as it is an immunological disease, what are the cells involved? CD4 T cells are involved. T lymphocytic cells are involved. So again this T lymphocytes on their surface they will be having some kind of receptors, right? So when this virus gets bounded to this receptor, it forms a bond. So once it gets binded to that, it will form a bond and then it undergoes internalization by reacting with certain enzymes like CCR4. So with that helps, it will enter into this host cell. Now this virus has entered into the host cell. So once it is entered into the host cell, what does it do? It will uncoat itself. So whatever the outer coat is, that will be removed. And then in core, what is there? There is two strands of RNA, right? So this RNA, with the help of RNA transcriptase enzyme, it will form single-stranded DNA. So again, the single-stranded DNA, using it as a template, another single strand will be formed, forming a double-stranded DNA. So this double-stranded DNA resembles the normal host cell DNA. So what does it do? Then it undergoes viral integration. So this with the viral DNA which is formed, that with the help of enzyme integrase, it will get integrated into the host cell DNA. So once it is in the host cell DNA, it undergoes replication. So during that process, multiple number of viruses will be produced. So when the presence of these viruses are there, what does our body do? It will show its immunological reactions by releasing certain cytokines and interleukins. So this is how the CD4 T cell destruction will be taking place with the help of these interleukins and cytokines. So this CD4 T cell which is having this viral DNA will be destroyed, and not only this, adjacent cells also gets destroyed, causing the destruction of the cells, or that tissue damage will be taking place. And also, so once they were destructed, what will they do? This virus particle which is present in the CD4 T cell, it will destroy the CD4 T cell, and also it will be released from this CD4 T cell which is damaged, and then it enters into the systemic circulation, causing the spread to different parts of the body. Okay, this is all about pathogenesis. So the stepwise will be discussing clearly here. So selective tropism for CD4 molecule receptor. So selective tropism means it has to bind to only a particular molecule, CD4 molecule receptor. So GP120, which is present on the surface of this virus, that will bind to this CD4 molecule receptor. Okay, and not only the CD4 molecule receptor, it also GP120 has also tropism or affinity to bind to this monocyte macrophages, microglial cells, epithelial cells of the cervix, Langerhans of the skin, and follicular dendritic cells. So it also has affinity to bind to these kind of cells, not only the CD4 T cells. So this is the first step, that is binding of virus to this host cell with the help of GP120 of the virus and CD4 molecule receptor. So these two will form a bond. And then internalization, as I already said, that other receptor called chemokine receptor, CCR, this will help in, and also GP41, these two will help in the internalization with the help of an enzyme. So with when this complex binds to this CCA4, so it will make the passage or entry into the host cell. And then uncoating and viral DNA formation. So once it has entered into the T cell, so with the help of reverse transcriptase, it will form a single-stranded DNA, and then using it as a template, and DNA polymerase will help in forming a double-stranded DNA. So now original RNA strand is destroyed. So here what happened? Actually in the virus there is presence of single-stranded RNA, right? But after that, it will be converted into single-stranded DNA and then to double-stranded DNA. So during this process, whatever the first initial RNA is there, that is destroyed. And during this process, it undergoes multiple mutations. So that is the reason there is no exact treatment for HIV. Why? Because as we are developing a medicine to one kind of pattern, again it's changing, and it's changing its mutations by mutations. So that is the reason there is no exact treatment for virus like HIV virus. And then viral integration. So this viral DNA of the host cell, it integrates viral DNA of this virus, enters into the nucleus of the T cell, and then binds to it and integrates into it with the help of integrase. So now this, whatever the host DNA and viral DNA together is forming as a single DNA virus, that we call it as HIV provirus. And then viral replication, and then it will undergo replication, this host cell DNA which is already having this viral DNA, both of them together will undergo replication. So once they undergo replication, more number of viral particles will be produced, so which will activate the CD4 T cells to liberate its cytokines. So Th1 cells will elaborate, will help in the formation of interleukin 2 and IFN gamma, interferon gamma, and Th2 cells will elaborate certain interleukins like 4, 5, 6, and 10. So these interleukins and cytokines, they cause the destruction of this of the CD4 T cell. And then RNA viral particles thus fill the cytoplasm of the host cell where they acquire protein coating. So these again, once these RNA viral particles again will be formed of these new viruses, so they will get these acquired coating proteins from these T cells, and again they will form the stable new viral particle which is having RNA and also the proteins, coating proteins. Then, so these released cytokines which were released, so these will cause the spread of the infection to other body sites, especially to the central nervous system by TNF alpha, that is tumor necrotizing factor alpha. And then viral dissemination, as I said, that whatever the virus particles that were released, that will enter into the systemic circulation, so that we call it as viremia. Septicemia means